All right, I'm going to start with the introduction. And by the time I'm done, it'll be 4:35. I think people have started rolling in. Thank you, everyone, for joining us for the CGIBD seminar today. It's always exciting to have one of our prior T32 trainees present because it goes to show the quality of the work that's being done and the quality of the applicants that we actually get and recruit to the program. Dr. Matt Foley is currently a postdoctoral fellow at NC State. And the work he'll present, I'm assuming, is going to be related to the work he did while he was on the T32 in the lab of Casey Theriot at NCSU. And he received his BS at the University of Rochester and then his PhD in microbiology and immunology at the University of Michigan, Ann Arbor, and has been a postdoctoral fellow at NC State since 2018. He joined our T32 program and was really instrumental in leading a lot of the discussions and innovation around gut biology and bacteria and the microbiota and how it basically relates to intestinal disease, specifically in the context of infection and how we can actually modulate it to impact infection. So Matt, the floor is all yours. Welcome.
Yeah, thank you for that nice introduction, Shazad. I'm gonna put this up. Hopefully that's coming through. Ooh.
Yes, it is. It is.
All right, awesome. Well, yeah, it's a pleasure and an honor to tell you about the work that I've been doing really for the last 3 to 4 years in my postdoc in Casey Theriot's lab in collaboration with Rodolphe Berengu. Technically, I'm now a staff research scholar in Rodolphe's lab, so no longer a postdoc. And I've switched projects, but this is still a story that we're looking to publish very soon. And so the story I'm going to tell you today is really about these Lactobacillus bile salt hydrolases and how we can potentially use those to manipulate not only the bile acid pool, but also leverage their activity to prevent or treat C. difficile infection. All right. Sorry. So I know members of the CGIBD are very familiar with the human gut microbiome. But I kind of want to explain it from our vantage point through our perspective. So this is a diverse and dynamic microbial ecosystem that harbors millions of genes that are not present within our own genome. And so within that genetic potential is really the capacity for the ability to generate hundreds and thousands of different molecules and chemicals that we as humans and as hosts wouldn't normally be exposed to. And so collectively, that metabolome is known to have a really profound impact on human health. And in the Theriot lab, we're really particularly interested in specifically the bile acid pool, which is the collection of these small steroidal-like molecules called bile acids. And these are host synthesized but bacterial modified metabolites, and they've been mechanistically linked to a number of diseases, including inflammatory diseases, cancers, metabolic disorders, and of course, infections from pathogens like C. difficile. And there we go. So how is it that this group of metabolites is able to influence so many aspects of our biology and our health? As it turns out, there we go. As it turns out, these small molecules are really form this bridge, this axis, metabolic axis between the host and the microbiome. So conventionally, bile acids are thought to sort of play a role in the digestion and absorption of fats and fat-soluble vitamins and nutrients, but really they have a much more profound role throughout our bodies. So they have a direct role in shaping our physiology by interacting with host-encoded and expressed receptors called bile acid receptors. The most sort of popular, most famous of which is something called farnesoid X receptor, or FXR. And so these bile acid receptors directly interact with bile acids throughout our body and regulate everything from inflammation to central metabolism and just general homeostasis. But bile acids also indirectly shape our physiology by interacting and shaping not only the composition, but also the function of our intestinal microbiota. So some bile acids can enrich for gram-positive spore-forming bacteria by acting as germinants. Other bile acids can, at the right concentration, select against certain bacteria using their detergent-like and weak acid-like properties. But this relationship between bile acids and the microbiota is really a two-way street, because members of the microbiota also have the ability to really shape the chemical complexity and diversity of the bile acid pool itself. So I'm going to walk through some of those transformations now, and it's going to get a little bit jargony, but I'm going to really recap and explain the exact important points of the slide at the end of it. So when we as humans make bile acids, we synthesize them de novo in the liver, and what we make is we consider them to be conjugated primary bile acids. So primary because we make them, and they haven't really been seen by any other bacteria yet, and conjugated because they're linked to an amino acid, either a glycine or a taurine. And so as these conjugated primary bile acids are excreted and then transit the intestinal tract, they encounter these microbial encoded enzymes called bile salt hydrolases, or BSHs. And so these enzymes have a simple function to remove and deconjugate that amino acid. Again, that's a glycine or a taurine. But it's absolutely a critical function, because these enzymes generate deconjugated bile acids, and it's these deconjugated bile acids that not only have an entirely different chemistry compared to their conjugated versions, but they also are precursors for a whole host of other microbial encoded transformations that really introduces the full chemical complexity of the bile acid pool of a healthy individual. And so, for instance, cholic acid can get converted into deoxycholic acid through 7-alpha dehydroxylation, which is the removal of this hydroxyl group into 7-carbon. And so as these secondary bile acids continue to transit the gut, they will eventually end up, there we go, in the terminal ileum, where the majority of them get reabsorbed and undergo enterohepatic recirculation, where they're brought back to the liver, they're repaired, they're reconjugated, and they can actually undergo this process several more times. Now, we are really the most interested in this upstream step that's encoded by these, that's carried out by these bile salt hydrolase enzymes, or BSHs. We really see these enzymes as sort of gatekeepers for the full diversity and function of the bile acid pool, because all of these downstream steps really rely on their action. So because of that, we have a lot of sort of basic fundamental questions surrounding their biology, trying to understand how they're used by members of the microbiota, but really what's their effect on the larger intestinal ecosystem, how they work at a molecular level. And for the purposes of this talk, I'm really going to hone this question down to really just ask specifically, how do Lactobacillus BSHs recognize not only, recognize their conjugated bile acid substrates, and how does that activity modulate and impact C. difficile pathogenesis? Now, we focus in on Lactobacillus as a genus, really because this is a good model genus. It's sort of a prototypical genus for deconjugative bacteria in the intestinal tract. They're a rich source of BSH diversity. They reside in the small intestine, where the majority of deconjugation occurs. So we really sort of rely on them as a model genus to understand how BSHs are used. And so in order to really survey the landscape of BSHs within the genus Lactobacillus, Dr. Sarah O'Flaherty, as well as a doctoral student, Echo Pan, in Rodolph's lab, first wanted to characterize some of the sequence diversity that's encoded within this genus. And so what they did is they bioinformatically analyzed the BSH ohm of lactobacilli by taking the about 4,000 lactobacillus genomes that are deposited within the NCBI, and distilling out from those about 2,000 BSH sequences, which they then reduced down based off of a 95% identity threshold into 85 clusters or groups of BSHs. And so I'm showing you a phylogenetic tree of those BSHs here, or those clusters of BSHs here. And so these names represent really the species that seem to dominantly encode for these groups of these clusters of BSHs. And what you'll see is around this phylogenetic tree is a few layers of interesting extra information about these BSHs and the species that encode them. So for instance, within this inner ring that's mostly blue, what you'll see is some information about the lifestyle of these organisms that encode these BSHs. Immediately what should pop out at you is the observation that these are mostly from vertebrate adapted organisms, meaning that and this really just sort of enforces this idea that these BSHs and really bile acid altering enzymes in general seem to promote the symbiosis and the commensalism of these bacteria with their vertebrate hosts. Now, in this inner level of red rings, what you'll see is the source of those strains. You'll find that a lot of these strains are isolated from humans, but also a lot of them are isolated from different animals. And so BSHs are not really just relevant only for humans and human biology, but really for many different hosts and even for animals that are of agricultural importance. And finally, in this outer ring, you'll see these tan wedges. And these wedges indicate BSHs that we were really interested in and wanted to better understand some of their functions in vivo. And so what we did is went on to heterologously express, purify, and characterize these BSHs in vitro. So I'm showing you here, and I hope you guys can see my mouse. I'm not sure if you can, but I'm showing you here on the left, this heat map is about 17 BSHs, which I decided to characterize and understand how they acted on a panel of conjugated bile acids. And so they're arranged here into their own little phylogenetic tree. And then in this heat map, what you'll also see is that these BSHs were surveyed for their activity on a panel of conjugated bile acids. So these left four bile acids are all glycine conjugated bile acids, and that's the G in front of their abbreviated names. And then the right four are taurine conjugated bile acids, and that's that T in front of their names. And what you can sort of immediately see or appreciate is that all of these BSHs really tend to prefer acting on either a glycine or a taurine conjugated bile acid. You can almost see a schism down the middle of this heat map. And what's even more so interesting is the fact that all of the taurine preferring BSHs are really confined to the single clade present here that I've highlighted in the blue. And so we found that to be really interesting, this sort of specialization for substrate preference. We wanted to better understand why or how mechanistically these BSHs are able to differentiate between these different conjugations. And so to do that, we really relied heavily here on some collaborators that we recruited for this study, and that's sort of hometown heroes for the CGIBD, Dr. Matt Redenbow, and then his student Morgan. And so Matt and Morgan are structural biologists and enzymologists, and they really were able to help inform our enzymological studies here. And so together we selected 6 BSHs from this tree highlighted in the red with these red arrows to better understand their structural biology and their catalytic mechanisms. So amazingly, Morgan was able to solve the structure of all 6 of these BSHs. So 2 of these are taurine preferring BSHs and 4 of these are glycine preferring. And just to give you an idea of how impactful this is to sort of BSH biology, there's currently 5 BSH structures deposited in the PDB. So Morgan's solving of 6 structures is really quite a feat. And so with these structures in hand, we can then really intelligently ask certain questions about their active sites and try and understand what it is about those active sites that dictate a preference for one conjugation type over another. And so Matt and Morgan's sort of seasoned eyes here were able to immediately point out a region of these BSHs that were of interest. And so showing you here an overlay of all 6 of Morgan's structures from the monomer of these proteins. And so what you can see immediately is the backbone of these proteins is almost invariant. It's very, very conserved. And there's this particular loop region in this loop that extends away from the core of the enzyme where the active site is, denoted by that C2, that second cysteine. And what was so interesting to them about this particular residue is the fact that at this site, at this location, there's this really nice stratification of a sequence motif between the taurine preferring and the glycine preferring enzymes. So here we see the taurine preferring enzymes all have this GVG or GTG motif, whereas all of the glycine preferring enzymes have this SRS or SRG motif. And what's even more sort of convincing about this particular region is, and what you may be asking sort of yourself, is that although this region, this motif, isn't located within the active site of the monomer of these proteins, it actually gets extended out into the neighboring monomer, so the dimer, and helps form the mature active site of the overall tetramer that is the BSH. And so what you can see here is the L-gas BSH-A. This is a structure that Morgan solved. This is a taurine preferring enzyme. That GVG motif on that loop, it gets injected into the active site and helps form and sort of caps off the active site of this BSH. And what you can see is that she was able to solve this particular BSH with a hydrolyzed TCDCA. So a hydrolyzed taurine was present within this active site. And you can see how it's really nicely accommodated by the valine that's present on this loop. And when we look at the glycine preferring L-gastride BSHB, you'll see immediately that when we look at that same loop, the arginine that is really conserved in this location would sterically hinder or clash with the presence of a large amino acid like a taurine in the same location. And so we formed this hypothesis that these the valine and the arginine between these two different motifs are somewhat dictating the substrate preferences between glycine and taurine preferring BSHs. And so we wanted to test that biochemically. And so what Morgan did is she tested the catalytic efficiency of the wild type versions of both of these two different enzymes, as well as a mutant version of these enzymes in which we've swapped those motifs, the GVG and the SRS, between the two different BSHs. And so looking here on the left at L-gas BSH-A, you can see this is a taurine preferring enzyme. It acts better on TCA compared to GCA. And when we make that mutation, we see that we lose some ability to act on both GCA as well as TCA. What's really interesting, though, is when we look over at L-gastride BSHB, this is a glycine preferring enzyme. And when we look over at the mutant in which we've swapped those motifs, we do lose some ability to act on GCA, but what's even more interesting is the ability, the increased ability to process TCA. And so this sort of helps convince us that indeed this arginine, this large side chain of the arginine, is maybe restricting the coordination of the larger taurine amino acid within that active site. And so we sort of concluded that these loop residues are playing some significant role in directing substrate preference, but do these preferences really even matter to BSHs? This is just an accident. And one way in which we think that they matter, or one case in which we think they may matter, is in the case of C. difficile. So I want to introduce C. diff now and sort of focus on that for the majority of the rest of the talk. But what C. diff is, so this is Clostridioides difficile. It's the causative agent of CDI, C. difficile infection. And it is a really common nosocomial enteric infection. It's a serious public health concern. And an infection is typically initiated when we ingest spores that germinate in the small intestine, which lead to vegetative cells then growing in the small intestine and the large intestine to high numbers, and where they then produce toxins that mediate the pathology of CDI. This typically happens in individuals who have taken antibiotics that perturb the intestinal microbiota as well as the metabolome, and that normally is there to provide colonization resistance from pathogens like C. diff. And specifically, we know that we often see in susceptible individuals a loss of BSH encoding bacteria, as well as accordingly the loss of deconjugated and secondary bile acids. And this is more than just an association, because we know that bile acids are able to to really regulate and govern the pathogenic life cycle of C. diff. So certain bile acids like TCA, for instance, are actually needed to germinate these spores into their vegetative and disease-causing form. We also know, however, that certain bile acids like CDCA are able to inhibit germination, and not just germination, but CDCA as well as other secondary bile acids can also inhibit the growth of C. diff, and as well as the ability of toxins to be expressed and act on our intestinal epithelium. So these bile acids really seem to regulate C. diff pathogenesis, virulence, and because of that, we wanted to ask the specific question of whether or not BSH activity could modulate the bile acid pool in a way that inhibits C. diff, germination, growth, and pathogenesis. So to answer that question, we first looked in vitro by using pure forms of C. difficile spores or vegetative cells in combination with either pure taurine, glycine, or deconjugated bile acids. And we first wanted to look and see if these various bile acids could differentially inhibit germination based off of the status of that conjugation on the bile acid. So I mentioned before, TCA is a very good germinant of C. diff germination. And so when we include TCA and incubate TCA with C. difficile spores, we're able to selectively plate those spores on media that allow us to calculate a percent germination, so the efficiency by which these spores are germinating, and we can set that to 100%. And then in addition to that TCA, we can supplement that with various bile acids and measure the ability of those bile acids to inhibit that germination. And so that's what the rest of these bars represent. And what you can immediately see is that while all of these, the addition of these different bile acids do inhibit germination to some extent, the red bars, which represent the deconjugated versions of these bile acids, are the most inhibitory, especially compared to the glycine conjugated versions, as well as the taurine conjugated versions of the same bile acids. Now, we also know that these bile acids can inhibit growth differentially. So I'm showing you here the minimum inhibitory concentrations of various bile acids with C. diff. So a higher MIC means that the molecule is less toxic, less inhibitory, whereas a lower MIC means that that molecule can inhibit C. diff at a lower concentration. And what I think you can immediately see really is that these taurine conjugated bile acids in the light blue are almost really not toxic to C. diff and seem to have a really high MIC, whereas in the red bars, you'll see those deconjugated bile acids are the most toxic and really are efficient at inhibiting C. diff. And so this sort of adds some fuel to the fire that these BSH generated deconjugated bile acids can be used to specifically inhibit C. diff. And we wanted to assay that using a really powerful set of samples that we have in the lab from a clinical study that was performed actually here at UNC Chapel Hill a number of years ago by doctors Michael Doherty, Sarah McGill, and Ajay Gulati. So this was an FMT study in which patients that were suffering from recurrent CDI, this is C. diff that's relapsed multiple times, are in line and are about to undergo an FMT. So this is a fecal microbiota transplant where you take the feces from a healthy individual and use that to treat the recurrent C. diff of a sick individual. And it's an effective strategy and treatment for recurrent C. diff. Now, before those patients received their FMT, a portion of their stool was sampled and stored. And so within this stool, we call this pre-FMT stool, is all of the metabolites, the microbiota, and really the conditions in which we imagine that C. diff is able to grow and successfully infect these patients. Now, what's nice about this is, of course, it's clinically relevant. These are sick individuals, but humans have a mixture of glycine and taurine conjugated bile acids. And really, that pre-FMT stool nicely approximates and mimics the in vivo colonic environment where C. diff would normally be causing disease. And now past members of the lab have actually studied these samples, and so we know a little bit about their metagenomics and the metabolomic profile of these samples. And for instance, we know that the abundance of BSH genes in these samples is almost non-detectable, so it's very little BSHs in these samples. In addition to that, the bile acid pool is completely shifted in a way that we would expect for a susceptible individual. So we see high levels of conjugated primary bile acids. This is a loss of deconjugated bile acids and secondary bile acids. So with these samples, what I wanted to do was an ex vivo assay in which I take that pre-FMT stool, resuspend it in PBS, and then treat it with a variety of BSH cocktails, either one that contains 3 taurine-preferring BSHs, one that contains 3 glycine-preferring BSHs, and then one contains the 6 BSHs encoded within those 2 different cocktails. And then inoculate those samples with C. difficile and then measure growth over time to see if BSHs can lead to the inhibition of C. diff growth. And what we did is look at this across 3 different recipients, 3 different patients, and we saw a lot of variability. And so looking at patient 1 or recipient 1, we saw that with each of those 3 different cocktails, we're able to significantly impact or at least inhibit C. difficile growth over the course of those 8 hours. You'll note, though, that the activity of the taurine preferring BSHs as well as the G and T preferring BSH cocktail were able to inhibit a little bit better than just the G alone. Now, recipient 2 was considered to be sort of a non-responder. C. diff really didn't seem to be affected significantly with the presence of any of these different BSH cocktails. But in recipient 3, we saw really robust inhibition of C. diff growth. And so this was seen across the 3 different cocktails that we tried. And so to validate these results and see, make sure really that these BSHs were acting in accordance with how we would expect them to act, we performed targeted metabolomics, looking at the bile acid pool of these samples after that 8-hour time period. And this was performed, this analysis was performed by Allison Stewart, PhD student in the Baker lab here at NC State. And really what we found was confirming in that the greater abundance of deconjugated bile acids that really seemed to associate with the greater the level of C. diff inhibition in those samples. And so that was really confirming, nice to see. And in fact, in recipient 2, where these BSH cocktails really didn't seem to be effective, we didn't see an increase in deconjugated bile acids. And so for whatever reason, this patient seems to be, the BSHs don't seem to be working in this particular patient. But I think that's really neat that it just reinforces the idea that we need these deconjugated bile acids to be there in order to inhibit C. difficile. Now, these samples are really great and they're really powerful, but we have a lot of questions about C. diff biology along the intestinal tract. And so these fecal samples were somewhat limiting in that regard. And so in order to address some of those other concerns and questions, we had to turn back to our mouse model of C. difficile infection. And so here we take C57 black 6 mice, wild type, that have colonization resistance to C. diff. And we give them antibiotics, cephalosporin, in the drinking water for 5 days, followed by a 2-day washout period. And this renders them susceptible to C. diff. Now, over the course of several weeks, that colonization resistance will start coming back over time. But we wanted to sample from several different time points over the course of the first week in order to see if the supplementation of BSHs could actually bring about the return of colonization resistance sooner. And so here, in order to sort of sidestep the need to orally gavage these mice with some sort of BSH or BSH cocktail in a different formulation, what we did here was a similar ex vivo approach in which we took these mice and we sacrificed them and then harvested their intestinal contents from various sites along the intestinal tract to individually ask how these BSH cocktails could be used to inhibit C. diff at various tissues. I will note too that so mice, unlike humans, have primarily taurine conjugated bile acids. And so we weren't too concerned with stratifying our cocktails based off of their preference, and instead really just used the G and T BSH specific cocktail here that I'm referring to as just the BSH condition. And what we wanted to test first was the ability of the BSH cocktail to inhibit C. difficile germination in the relevant intestinal compartment, which is the small intestine. This is where C. diff is normally thought to, which is where their spores are normally thought to germinate and sort of initiate infection. And so across these various time points at days 0 through 7, what we found was that the ability of C. difficile to germinate was significantly reduced and inhibited when these small intestinal contents were treated with the BSH cocktail as opposed to just the PBS buffer control. And so this was really confirming and promising, but C. difficile growth is also a huge metric that decides the pathogenesis of this pathogen. So we also wanted to look at the ability of this BSH cocktail to inhibit C. difficile growth at these various different sites along the intestinal tract. So within the small intestine and the cecum, we did see significant inhibition of C. difficile growth, although to different extents. So within the small intestine, where bile acids are at their highest concentration, we saw a significant and robust inhibition of C. difficile growth, and in fact, in some cases, complete killing of C. diff. Whereas in the cecum, this level of inhibition was much less pronounced. It was really minute, but it was somewhat significant. And so we've concluded that these BSH dependent effects for C. difficile inhibition are going to really depend on the concentration of bile acids at the particular site that they're acting on, as well as of course, the site that the tissue that they're working in. But we also wanted to understand what types of bile acids these BSHs were really acting on. And so again, we performed targeted metabolomics on these samples, and I'm just going to show you small intestinal samples here as an example. And this is again performed by Alison Stewart in the Baker lab. We found some really nice sort of trends in which the BSH cocktail was able to decrease the level of TCA, the conjugated version of cholic acid, and produce the deconjugated version cholic acid in almost a stoichiometric amount. So the exact same trend with TCDCA and CDCA. So this was, again, really validating, really confirming that BSHs are producing these deconjugated bile acids that inhibit C. difficile growth, and we can show that in vitro. But we were really surprised to find that there were some other bile acids that were present in these samples that were really interesting. So I'm showing you here the detection of 3 different types of conjugated bile acids, versions of cholic acid that are conjugated to either isoleucine, phenylalanine, and tyrosine. And so one, it was really interesting that we were finding these completely different versions of bile acids. I know up to this point, I've only talked about glycine and taurine conjugations, but more so, it was really interesting that we were seeing these in the BSH condition in which we've treated these samples with this deconjugative enzyme. So what are these molecules? So canonically, I'll take a step back. Canonically, bile acids are conjugated in the liver to glycine or taurine. And then while in the intestinal tract, those conjugated bile acids, again, are deconjugated by BSHs and coated by the microbiota. Recently, about 2 to 3 years ago, I guess 4 at this point, amazing how time flies, there was a discovery of these novel bile acids, what we call non-canonical conjugated bile acids, which are bile acids that are conjugated not to glycine or taurine, but rather to all of these different amino acids like phenylalanine, tyrosine, leucine. And this was really interesting. This is really something that sort of shocked us because for decades, glycine and taurine were thought to be really the only amino acids that we could find bile acids conjugated to. What's even more interesting about these molecules is that this conjugation isn't performed by the host in the liver, but rather by members of the microbiota. So this is another mechanism by which members of the microbiota are, in their chemical complexity and their enzymatic diversity, is increasing the diversity of the metabolome in our intestinal tract. Because these molecules are so new, they really haven't been studied to the full extent. We still don't totally understand what their role is in maintaining physiology and shaping our health. We do know that they likely have the capacity to do so. So some of these bile acids are really strong agonists of FXR. Once again, this is that bile acid receptor that's really a master regulator of so many different aspects of our physiology, including bile acid synthesis, but also inflammation and metabolism. And you'll note that, for instance, tyrosine conjugated cholic acid is almost as strong of an agonist compared to the synthetic pharmaceutical called obeticholic acid. Finally, what's interesting about these non-canonical bile acids is that it wasn't observed that they were being able to be deconjugated by members of the gut microbiota. So potentially, these are more recalcitrant compared to just their glycine and taurine counterparts. Well, that really got us curious, and we really wanted to understand the role of these different bile acids as they relate to BSH biology and as they relate to C. diff. And so we reached out to the Dorstein lab, which had originally reported finding these molecules, and we got in contact with a postdoc there, Dr. Emily Gentry. So she's in the Dorstein lab at UCSD, and she's an organic chemist. So she's taken this synthetic chemistry approach to understanding this whole group of molecules. And so what she's done is actually synthesize hundreds of these molecules. So all of these different bile acids, she's synthesized them to 22 different amino acids, excluding glycine and taurine, in order to synthesize this pool of bile acid amidates that she can then use mass spec to collect spectral data from these different molecules, and then use that spectral data to essentially search for these different molecules within publicly available metabolomic databases. We can see where and when we find a lot of these different bile acids. What she's found is that many of these bile acids exist in the wild in various organisms. They do differ in their abundance based off of their conjugation and based off of the type of bile acid that they're linked to. But there is this whole family and field and pool of bile acid amidates that exist that really up until a few years ago, we as biologists and microbiologists had not appreciated. And so this was really exciting to us. And of course, us studying BSHs, we really wanted to know if these different conjugated bile acids could be deconjugated and processed by BSHs. And so she gave us purified versions of those non-canonical conjugated bile acids that we identified from metabolomics, isoleucine, leucine, phenylalanine, and tyrosine conjugated cholic acid. And we screened for for activity on these substrates with 11 of these different BSHs. And don't let this heat map fool you. So the scale here goes up to 10 for this particular set of data, whereas the heat map that I showed you in the beginning of this talk, it went up to about 1,000. So these enzymes are about 100-fold worse, at least, compared to glycine and taurine conjugated substrates. So these seem to be really recalcitrant and really poor substrates for BSHs. But we're really only looking at a narrow subset of all of the different types of conjugated bile acids that exist. And so we really wanted to ask whether or not BSHs could act on some of these different non-canonical conjugated bile acids and shape that sort of non-canonical pool of bile acids. And so to get at that question, we wanted to take this high throughput approach. And so using Emily's mixtures of these various bile acids, what she had done is react individual bile acids like cholic acid, Ca, with a pool of amino acids, so 22 different amino acids, excluding glycine and taurine, and then relying on Alison Stewart's ability to separate all of these molecules out using LCIM-SMS. We wanted to take this approach in which we reacted individual BSHs with a pool of non-canonical conjugated bile acids, and then over time measure which ones within that pool, which of those molecules are able to get deconjugated and then also see which of those are not able to be deconjugated. And throwing a lot of data at you here, I'm showing you the results from that study. So we took 5 different BSHs and reacted them with 4 of these different pools of non-canonical conjugated bile acids. And so what you can see is that sort of immediately is that there is a lot of variability when it comes to the type of the BSH, the type of the bile acid, cholic acid versus chenodeoxycholic acid, and then also the type of the conjugation. So whether it's alanine, which is really well processed by many different BSHs, or for instance, arginine, which is a little bit more recalcitrant and is not as easily processed. We saw so many different trends, and I'm really not going to get into some of the details here, but we did find that one particular BSH from Ellen Gluvier, we call this L-ing BSH, was a particularly good deconjugator of a lot of these novel conjugated bile acids. So this is not a trait or a feature of all BSHs, but it may be a feature of some. And that's something that, moving forward, I think we really want to think about when deciding how to rationally manipulate the bile acid pool. Now, moreover, this is really interesting, but does it matter to C. diff? And so we wanted to know whether some of these novel and non-canonical conjugated bile acids could actually impact C. diff biology. And so growing C. diff in the presence of some of these non-canonical conjugated bile acids, we found that, interestingly enough, some of these were actually able to enhance the growth of C. diff, although it's a little bit subtle. But a lot of these bile acids were actually able to also inhibit its germination. So using the same type of germination inhibition assay that I showed you previously, you can notice that not isoleucine conjugated cholic acid, but tyrosine and phenylalanine conjugated cholic acid were able to significantly inhibit C. diff. And this is these molecules are found at their highest concentration within the small intestine, where we would expect C. diff to be germinating. And so we think this is a really relevant readout for how they may be impacting C. diff in the wild, or at least in vivo. So we know that some of these non-canonical conjugated bile acids can impact various stages of C. difficile life cycle, but we really have a lot of questions remaining concerning how they impact colonization resistance and the larger microbial ecosystem that is the intestinal microbiome. So really, to conclude today, I've told you a lot about how BSHs can direct their substrate specificity to the conjugated amino acid of different bile acids. I've also showed how deconjugated bile acids, so the products of BSH activity, can be strong inhibitors of of C. difficile pathogenesis, germination, growth, etc. And I've also talked about how these BSHs can act variably on these various non-canonical conjugated bile acids. Now, moving forward, we really want to better understand that relationship between this whole new set of molecules and how they impact C. diff, but also how they impact so many different aspects of our physiology and our health. And so that is certainly something that we're going to be striving for. But we also want to take this a step forward and make this translational and be able to deliver these BSHs in vivo to see if we can actually inhibit the severity of CDI, restore colonization resistance. This is something that we may consider to be a quote unquote enzybiotic, and something that is able to restore colonization resistance without the complexity and the heterogeneity that is a fecal microbiota transplant. And so with that, I do want to thank a lot of people, because this was an incredibly collaborative study that we did. And so, of course, I want to thank the Terrio Lab and all of its members, past and present, especially Casey Terrio, who's been a wonderful postdoc mentor for the last 4 years here at NC State. The Berengu Lab, Rodolph, Sarah, and Echo have been a tremendous support system in this study and have been really experts in probiotic biology. The Baker Lab has provided us with the tools to analyze all of these complex metabolites. And so Allison has really done a fantastic job at mastering the separation and analysis of these different metabolites. The Dorstein Lab at UCSD, especially Emily, we really wouldn't have been able to study these non-canonical conjugates without her ability to synthesize them. The Redenbaugh Lab was a huge a source and contributor to this study, especially Morgan, with her ability to perform these enzymological assays and her structural biology skills. And then finally, also thanking the gastroenterologists that played a role in collecting those FMT samples, my CGIBD committee, and some of the funding that we've had over the years. And so with that, I'm happy to take any questions. And yeah, I look forward to the conversation. Thank you.
Matt, thank you. That was incredible. Congratulations to you. And mind blowing for sure. We have questions coming up in the chat, but I'm going to just ask one of my questions really quick because I'm sure that it's naive. Non-canonical bile acids, man, that's fascinating. Can you actually go back and look at the metabolomic data sets that have been produced on IBD to find them? Have people been ignoring this? Is there any way to go and really find what their role might be in potential disease? I'm just curious, can you actually do that, or is it naive?
No, it's not naive at all. And yes, it can be done. And in fact, it is being done. And so study, there's a preprint out right now. I'm happy to send that to you by Emily Gentry, the lead author on that study, has in fact done that and started looking at the association between these non-canonical conjugates that she was able to detect and particular, IBD cohorts. And that's the great thing about these metabolomic data sets is you collect so much data and only some of those spectra you're able to assign metabolites to, but all of the remaining sort of dark matter still remains. And so you're able to years later go back and actually assign molecules to those spectra and understand what those molecules are. And so I think that's sort of a fascinating approach to biology in general.
Because we just got metabolomics data back from over 100 IBD patients in control. So I'm gonna email you in a bit and we'll chat. There's a question in the chat by Dr. Anderman. She said, beautiful talk, I agree. What will be the pros and cons of in vivo delivery of BSH versus in vivo delivery of, say, a deconjugated secondary bile acid? And can we use urso deoxycholic acid to inhibit C. diff since it's already available clinically? Sorry, Tess, I asked your question. You could have asked it yourself.
No, thank you. I appreciate it.
Yeah, that's a really great question, Tessa. So let me get at different parts of that. So there are some benefits to strategic benefits to delivering the enzyme as opposed to the actual product itself. So the product being bile acids. So because the majority of bile acids get absorbed in the terminal ileum and undergo this intrahepatic recirculation, when people sort of gavage with these different bile acids, in order to successfully get them to the large intestine, you really have to overwhelm the system. And so you get sort of a dosing issue in which you have to add a lot of bile acid, and that alone can have negative effects and negative consequences. And I'll just remind you too that C. diff, a lot of its disease is caused in the large intestine as opposed to the small intestine. So really, it's the large intestine that we want to target more than anything. And can we use UDCA to inhibit C. diff? Yeah, I think we can. So Casey Terrio has her postdoc or her grad student in the past has looked at some of the effects that UDCA has had on the pathogenic life cycle of C. diff, as well as the host response to C. diff. And in fact, the postdoc Colleen Pike, who I believe is on the channel right now, is continuing those studies. And so no doubt she's found a lot of interesting things there that further answer your question. But yeah, I think you're right on the money there with that strategy.
Thank you so much for that.
I'm gonna keep going through the questions. You got a bunch. So Balfour, I'll get to definitely. Dr. Matthew Tillman asking about looking back at your BSH structures. It looks like mutating the valine to a serine would allow for a polar hydrogen bond and interaction between enzyme and taurine groups. Could you explore engineering the enzyme to better deconjugate TCA? Yeah, it's an interesting observation. Definitely. So I don't think that the motif that we looked at is necessarily the only motif that decides activity or substrate preference. I think there's a lot of optimization that could go into either making better acting BSHs or more specific BSHs. And yeah, absolutely. I think there's a lot that can be done there. And really in the field, there has not been
too much done regarding substrate preference, understanding and engineering the substrate preferences of these enzymes. So it's certainly of interest to me.
Joseph Burkloff, another T32 prior trainee, wants to know, is it known which species of microbes makes these non-canonical conjugates? Would it be possible to administer specific prebiotics to increase these species?
Yeah, well, thanks, Joe. Hey, Joe. We still don't understand how they're doing this process of reconjugation. But to answer your question, so many bacteria in our microbiota are doing this reconjugation, which is really almost the most mind-blowing thing here, because it's actually so widespread. And so there's 2 studies out now that have looked across different HMP strains and have screened for the ability of these bacteria to do it. It's not just gram-negative, or it's not just gram-positive bacteria, it's gram-negative. It's all of these different, and they do it variably. So not all bacteria are created equal here, but so many bacteria seem to be performing these transformations. It's really mind-boggling. And just as an aside, it's just so amazing that we haven't figured this out before, because this is almost like a conserved function among a lot of our commensal species.
But that means there's no one silver bullet.
There's no one silver bullet, although with that being said, a lot of these do this at a very, very low level. So it would be amazing to find a bacteria that does that extremely efficiently. And I think you could probably maybe, once we figure out the mechanism behind that, engineer it and augment it. But as of right now, we have no idea.
That's like
amazing talk.
Yeah, perfect significance for a grant, right?
Yeah. Oh, yeah.
That question. Miriam Ock wants to know whether, I think it's similar to what Joseph asked about deconjugated bile acids or BSHs are inhibiting other strains of C. diff.
Yeah, so we have not looked here at the ability. So I only used a single strain in these studies. This is a clinically relevant strain of C. diff called R20291. So somewhat limited in scope here. We know, and there's been studies done that have looked at various clinically relevant strains of C. diff, that many, of course, there are some strain-to-strain variation and differences, but really in a conserved way, C. diff is uniquely susceptible to a lot of these secondary and deconjugated bile acids. And so we know kind of from the literature, and a lot of this work's been done previously by Casey Terrio, that C. diff is sort of uniquely and exquisitely sensitive to alterations in these bile acid pools.
Excellent. And last but not least, Balfour.
Yeah, I apologize. I got in very late because I just made it up to the cabin and I was able to connect. But 2 clinically relevant questions. Number 1, how important is germination of spores in the continuation of an infection once it happens? You made the comment, small bowels, kind of the dominant place for germination to occur. But is it important in sustaining an infection? How much is just replication versus sporulation, regermination? Clearly important in recurrent C. diff, but how important is it in just continuation of a ongoing infection?
Yeah, that's a great question. I don't think it's very important for primary infection. So if you are to be infected by a single spore that germinates and then expands out, I don't think that, I think that would be sufficient to cause severe disease. I think, you alluded to that maybe during recurrence and recurrent C. diff, the ability of C. diff to create these spores that may be sticky and stick around literally in the gut and then germinate at a later time period and come back after antibiotics are gone. That may be a mechanism by which C. diff does establish recurrence, but I don't think that the continued ability of C. diff to sporulate and then germinate while in one person's intestinal tract is important.
Right. So your therapy, deconjugation therapy, would not be amenable to hastening recovery. The second question that kind of is a spinoff of that is many Clostridia, including beneficial Clostridia, are spore formers. the Nature paper by Kenya Honda used spores to actually identify the protective bacteria. How important are deconjugated bile acids in preventing germination of helpful Clostridia spores? And is there any specificity that you're aware of?
Yeah, as far as I know, TCA, so taurine conjugated cholic acid, is really the bile acid that people use when they want to try to induce spore germination of all Clostridia, really. I'm not really sure why that is a conserved trait. I'm not sure if there are other Clostridia or spore formers that exist that preferentially like other bile acids as germinants. I don't know if that's been looked at, but yeah, it really seems like TCA is important. Now, are these other deconjugated bile acids able to inhibit spore germination for these other species? I'm actually not sure. That's a great question. I would assume that a lot of these deconjugated bile acids are able to inhibit not just maybe germination, but also the growth of normal commensal species too. And so it's really something that we view it as something that needs to be dialed in. So we don't want to come in with BSHs and deconjugate every bile acid, but rather in a healthy individual, there's some low level or maybe medium level, I'll say, of deconjugation just constantly occurring. And we want to be able to mimic that and replicate that with our BSHs. And so that's one of the challenges. But yeah, we do think that there could be.
You may be aware that you may be aware that TCA and glycine conjugated bile acids are increased in IBD patients, and I wonder if that might be a susceptibility for C. diff infection in those patients. The relative abundance of TCA versus CA, for example, conjugated, deconjugated, is increased toward the TCA and particularly in IBD patients.
Yeah, absolutely. Yeah.
Great talk. Fantastic.
All right. Thanks, Belfer.
You did the T32 proud.
Thank you. Appreciate all you've done for me.
Anyway, great work, great questions. It's exactly 5:30. I think on that high note, I think we should probably call it a evening. Thanks, Matt, for your time. It was fantastic.
Yeah. Thank you, everyone.
Thanks, Casey. Appreciate it. Yeah, bye.